Hi everyone, my name is Kevin Moyung, and I'm a PhD candidate at Duke University, co-advised by Dave McAlpine and Alex Hardemink. I'm interested in understanding how genes are regulated through the interplay of chromatin and gene expression, and our group's focus is to study this mechanistically through the lens of chromatin occupancy. Studying the gene regulation has traditionally been done through expression-based studies, but today I'm going to show you how chromatin modulates transcription, how it can recapitulate existing gene regulatory networks, and also reveal potentially novel regulatory interactions not found in prior studies. In Figure 1, we profiled the chromatin occupancy of 201 mutant yeast strains, where each strain had a transcriptional regulator deleted. Using MNA-seq, we were able to obtain the occupancy profiles of nucleosomes and transcription factors genome-wide. To validate our approach, we looked at the chromatin dynamics associated with the galactose metabolic pathway in yeast. In the presence of galactose, Gal80 acts as an inhibitor and prevents Gal4 from binding to and activating the Gal genes. We can stop Gal80's activity by deleting it in our yeast strain, and as a result, Gal4 is shown to bind to the promoters of the Gal genes and disrupting the nucleosomes. The plot in 1C shows the MNA-seq fragments plotted by their fragment length versus genomic position. Lengths of around 150 to 200 base pairs are nucleosomes, and fragments with lengths below 100 base pairs are deemed to be transcription factors. In Figure 2, we obtained the genome-wide landscapes of the chromatin occupancies for all of our mutants. In this example, we're looking at the MET10 locus, and in a wild-type setting, there's a transcription factor bound to the promoter region of this gene. However, when we delete CBF1, we see an absence of the TF occupancy. After looking at the motif evidence here, it turns out that this transcription factor is indeed CBF1. And you don't see this happening in any of the other mutants, which shows that CBF1 effectively regulates MET10. To quantify and summarize the chromatin dynamics, we used the Jensen-Shannon divergence metric to calculate the differences in chromatin profiles. In Figure 3, we are showing all of the CBF1 binding sites in the mutant compared to the control. On the rightmost plot in 3b, we're showing that the JS divergence effectively summarizes chromatin changes that are occurring as a result of the deletion of CBF1. To see if we could recapitulate gene regulatory networks with our chromatin data, we wanted to see if there was a relationship between chromatin dynamics and transcription. It turns out that many genes that have significant chromatin changes also have significant expression changes associated with them. In Figure 4, we're plotting the log2 JS divergence versus the log2 expression fold change. Points colored in red are genes that have both significant chromatin and expression changes associated with them. And in Figure 5, we wanted to see if there are consistent patterns between upregulated and downregulated genes. In 5a, we found that most of the upregulated genes had an increase in transcription factor occupancy, whereas the downregulated genes showed the opposite occurring, with a decrease in transcription factor occupancy across those genes. Focusing on the BAS1 mutant in particular, we found that most of the downregulated genes had a loss of BAS1 binding associated with them, which is what we expected. Finally, seeing as we are able to reconstruct a gene regulatory network from chromatin, the ultimate goal of our project moving forward is to expand existing networks with our data, which provides a much deeper and more mechanistic context into gene regulation. Thanks for attending my poster presentation, and I'm happy to take any questions.